Okay, hello everyone, and I think we may start very soon. Let's just give a few more minutes to anyone who would like to join us. And to all the participants, please don't request to go live. This is my stage for today, so you're here only to listen. Maybe interact with me a little bit, but I'm the live. Oh, that's what happens. I'm sorry. You have to turn everything down. Лилия, пожалуйста, перезайдите, потому что я случайно разрешила вам выйти в эфир, и это не лучшее решение.
Хорошо.
Yes, please, let's be very cautious about what the buttons that we push. And okay, I think it's right time for us to start. So if everyone or anyone will join us later, they're very welcome to do so. And I can share a few of my slides. Yeah. So a bit of spoilers, but we'll go to the first one. All right, so why don't we start? So today's topic is data analyst in medicine and life sciences. Sounds very exciting, and nevertheless, it's a very broad topic. So we will see what we could cover today. I hope we would cover a lot, but we will see what we'll manage to do. All right, so I'm very sorry for any inconveniences, but I'm not really familiar with the system of MTS-Link, but I will do my best to provide the best experience for you. So, all right, I'm your host for today. I'm Julia, but I will talk about myself a little bit later. And for the beginning, why don't we start with something very interesting and shocking, by the way. So if you look at this slide, and it would stop moving, I hope it does. So what is the shocking fact? That actually 80%, this is how similar protein coding genes of mice to human. Isn't it fascinating? I think it is. But humans are larger, smarter, and live longer than mice, right? Not to mention that humans don't have fur or tail. So then how does it happen? Why it is that? The reason we have been able to calculate our similarities to mice is because the massive effort of sequence of genomes of humans. It's called Human Genome Project, as well as sequencing genome of mice and many other organisms, by the way. Sequencing of genome involves reading through A, C, T, Gs, and that make up our DNA, basically, and DNA of any living organism. Does anyone knows what stands for these four letters? It actually do have something by this name. So if you know something about that, you can share it in the comments. We will read through that later. On what else we could say about that? Anyway, it has gave us a lot of information what our genes are, how are they organized, and how it has helped us as humans, how we diagnose the treat of human diseases. Does it even help? Well, it actually does a lot. But genome sequencing involves generating very large sets of data, like very large. So we rarely need to very powerful tools to read all this A, C, T, and G's. And finally, that's where the field of bioinformatics comes in. That's why we're all here today, right? So extremely powerful computers are being used to store and manipulate all of this data, right? And people behind computers are the stars that we're going to talk today are bioinformaticians. And there are also scientists who are often trained in both biology, math, and even computer science. And sometimes even other broad areas come in. So they develop methods, software tools to program computers to dig through and make sense of the data. Does it sound anyhow familiar to you? Isn't it what any data scientist or researcher does? Well, you may say so. I would not disagree with you. But what makes bioinformaticians so unique? So, how does bioinformatics help us learn what humans and mice have 80% similarity in protein coding genes? By the way, for someone who doesn't know, our genome is very large, but actually the part that makes the most sense is the protein coding genes. They make up just around 10% of the whole genome. This is also a fascinating fact. And don't judge me on numbers, it's just a rough calculation. But that's what, when I say gene or genome or anything like that, most likely I would speak about protein coding genes, all right? One way bioinformaticians approach this type of question is looking for small sequences in one genome that match the other genome. Like, let's look at the picture. So we see the lines of ACTG and so, and they might match with each other or they might not, right? But this is somehow very similar to doing a word search on your computer. But instead of looking for, I don't know, a word Pushkin in your Russian book collection, it scans for AT, GCA, GGT, CTA. This is a random combination, so it's not what we always look for, but this is just a part of genome that we try to match. So this is our small sequence that we try to catch. So once matching area is found, we design an algorithm that scans past both ways, both ends, right and left. Well, we don't exactly know what is right and left, but it just means we try to find something that goes prior this and after this. Oh my God, this is so much moving. Sorry for that. But as I said, I'm not familiar with the system, so we will take it as it is. Too many spoilers for you, but I hope you don't lose your interest too fast. Let's come back to the slide I was talking about with our sequences. All right, here we go. I hope it won't move. All right, so what do? We do scanning from both sets to find what are we looking for. We are looking for either gap, so it would be a mismatch or a match, right? To see how far the matching will extend. In this case, do the letters after CTA continue to match between the mouse and human sequence? If they do not, and the difference is present, we can analyze the following sequence and start to figuring out what genes are involved in many of traits that makes us human very different from a mice. Do you see how it goes? Do you see how it works? So if you have so much similarity between human and mice, shouldn't we be very similar as organisms? Well, we basically are, as we are kind of mammals, but we're also very different. And the thesis and the theory is that something that makes us different is coded in this mismatching sequences, right? Does it make any sense to you? I hope it does, because it's practically how it does work. So if we find the difference, so we hope that this difference is coded by this unmatching sequence, right? So to figure out how genes are involved in many traits that make us humans and mice different. What do we have in different? Likewise, I said it might be very many factors: size, fear, tail, but at least it's brain development, right? So I do consider myself a little bit smarter than mice, but just a little bit. So the flip side of this is that we can also find sequences that are highly similar across the species, and it's not only about humans and mice. It also goes to many other organisms. So humans have genome in common with plants, flowers, and even some bacteria. And I'm not joking. So this kind of genomes, and I would say it's regions of genomes, they are called consort genes. Why do they call them that? Because they're very conservative, and this is actually what's stuck in many organisms that are alive for today. And because they're shared across so many species, they likely code for some very essential genes. Something that helps you to breathe, to stay alive, to feed yourself, to survive in the nature and in the wild. I hope it also does make sense to you. So what else could we add? So besides that, in addition, if finding similarities and differences between two genomes of different organisms, sequencing technology has also allowed us to pick up difference in DNA between different people. So now we're talking about difference between me, you, and somebody else. So we're now working with the humans. So this has been particularly important because it helps us better to understand human diseases. What kind of human diseases? Well, basically, vast majority of them. It's not like flu, right? But it's something most likely to be genetically associated with some genomes or genes that we are looking through our sequencing. So if it's a diabetes or cancer or any other crucial disease, we would probably find something that would help us understand this disease. So let's say that species base in genome was different from person to person. Base in gene is just a letter or a combination of those letters like A, T, C, Gs, as I mentioned earlier. You can also see it on the slide. So we found that, and you want to see that difference way important for disease like diabetes, for instance. What can you do about that? How do you find if this essentially important or it's nothing to do with that? You can sequence the base in 50 healthy humans and 50 patients with diagnosed diabetes. This is very important, diagnosed diabetes. If 47, for instance, people with diabetes had an A, while only 5 out of 50 healthy people had an A. What does it mean? What conclusion will come to your mind? Well, basically, this would be a strong evidence that there is association between A variant and the disease. Of course, there are multiple questions that would arrive with that, like, is it statistically important and valuable found? Well, this is the job that we would also do as bioinformaticians or any kind of data analyst, all right? But for simple example, let's keep it so. So we found that this variant actually stands for the difference between healthy people and someone with diabetes. But importantly, it could not mean that variant, this letter A in this specific position caused the disease, right? Because there's so many factors that actually cause the disease. So what we say then? What we say about that? Do we know the difference? Well, we do know the difference, but the difference is it's associated with disease, but it is not the cause of disease, probably. I hope we could explain what's the association and the cause-reason link is. I hope you know the difference. So, in simple words, association, almost like correlation, it does show that these two factors do interact with each other, but it basically does not prove that one is cause of another one. I hope it's also understandable. All right, so researchers has developed the technologies that are able to look across hundreds of thousands of sites across genome for this kind of single base differences and have looked for correlations between certain base and disease. That's what I mentioned. These experiments are known as genome-wide association studies. This is very speaking for itself naming, all right? So once again, what do? We'll look through our sequences. For now, it's just a single base difference. Well, this is a huge simplification of what we do as researchers, but it's very good showing example. So we do look for these single base differences to see if there are any associations and differences between someone healthy and someone with diagnosed disease. All right, we'll be staying there. Okay, so what do we have next? So we found some associations. Is it the answer to all of our questions, even though if we're looking not for single base, but for multiple base differences? Well, not exactly. Let's talk about that. Oh my God, stop moving. Yes, I hope it would stay here, stay still. So, sequencing and bioinformatics, bioinformatic analysts, and any other researcher are also becoming increasingly important in modern world for diagnosing and treatment of cancer. This is one more topic to talk about because cancer cells are often having many mutations and changes in nucleotide codes to patients' normal cells. So we see that we don't only look for general difference, we also look for mutations that might occur or not occur in different types of cells. So now we're looking for specific cells. As you know, cancer cells are often so dangerous because they do duplicate themselves too many times rather than healthy cells in a human body. So that's the problem. We cannot control how they reproduce themselves. And that's often caused by some mutations that happen to be in those cells. How do we define those mutations if everything in us is a part of the genome? So we actually look to the genome and we try to find the genes that are probably the cause or associated with these mutations. All right. So as we can learn more and more about human genome, cost of sequencing decreasing. It also comes with new technologies and as just time passes by. Doctors can order sequences parts of their patient's genome for relatively low cost. I would say relatively because for us as individuals, it still would be quite expensive. If you have ever come across any advertisement of, would say, any genomic passports or anything that would let you define your ethnicity, it still costs you some money. And just imagine that this company doing thousands of that researches and they do some end of the sequencing so they could lower their costs. But for someone individual doing that, this is very expensive still. But there are many companies that do provide the services, so some doctors, hospitals, or research centers could do that for their patients. So by using the algorithms to compare patients' tumor cells to the normal genes, as well as to the tumor of many other patients. So now we do not compare only cells or genomes of one person to himself, we also compare these tumor cells to cells of another human being. So we would like to see a wider range of comparison to see if there is anything statistically important that would lead us to something that we could call at least association. Wait a second. Yeah. So doctors can quickly pinpoint the changes in the DNA that are causing cancer cells to grow uncontrollably. This helps them choose the best treatment for their patients. This is also important if we talk about the cancer, and I believe this is not the slide for that. This one is not even, yeah, this one. So for tumor itself and the cancer, there are so many types of that. So the treatment would differ from one type of cancer to another one, which actually leads us to the need of understanding every type of cancer itself. So we need to sequence many genomes for that, right? So we need to do our prior work to make that even countable. All right. And by the way, if there's anyone who uses this system a little bit often than I do, and you could advise me how to stop scrolling the slides without me, I would appreciate that a lot. But I hope it's not bothering you that much. So what else is bothering me is as our ability, oh my God. I think I need to sequence the genome of my laptop. Why it's so moving fast without me even doing anything? But unfortunately, I don't think my laptop has a genome. All right. So as our ability to sequence genomes continue to increase, in bioinformatics will need to contain and develop faster, more advanced algorithms, right? Because we need to handle this massive of data. An important part of this field has been a development of long centralized databases of genome sequencing that can be accessed by anyone who needed, a doctor, a researcher, or informatician, a statistician, whatever you can call it. And the challenge for the future is to continue grow these databases in a way that helps scientists to do important new discoveries and presenting the primary privacy of the patients. And you have to remember that actually the genome data also considered as privacy data. That leads us to many more questions about how to store it, how to research it, and how to share it with a patient to patient or with some group of patients that are not being like sequenced for this exactly research. And let's move further. And I hope you had a wonderful introduction. I did my best to explain it as simple as I could. And even though I might have failed, I hope you had fun. So I think it's time for me to introduce myself a little bit. So I don't want to spend too much time on that, but there is a few topics I would like to cover. So as you see, I do have quite of experience in working and also in studying. I have a bachelor's degree and a master's degree, and one of them is actually in data analysis in biology and medicine. So I could call it my real passion, if you want. And by the way, if you had mentioned, this is a protein 3D structure behind me, so I hope you enjoy my background as much as I do. So what else is interesting? Well, there are many ways how you can get in bioinformatics or any related field to that. And I would say my path was very, very typical to that. So I had my very basic first education of bachelor's in mathematics and computer science. And then I realized, so I was really looking for something to implement my knowledge. And I did knew by that time already that best community, best tasks, and everything exciting is always on the edge of science and edge of production and edge of multiple fields. So I was actually considering two options, as you say, it's finance and medicine. And I was looking for some factor that would shift me from one field to another and like help me to make my choice. And here it came. So I was attending the first summer school in machine learning and bioinformatics from actually High School of Economics. And the head of the school was in future the head of my master program. And this was very fascinating summer. So it was online, and we did have a lot of speakers coming from various backgrounds and coming from abroad. So it was a unique, very, very cool experience. But rather than that, what I did remember from the school that I did make my mind after that, but something even more important, that I didn't understand the word these people were saying. So they were talking about sequencing, sites, genomes, proteins, structures, high-C data, RNA, and many other things that I have never heard of. I think I had my last biology lesson like 10 years prior to school, and I was always very physics, computer science, and mathematics myself. So I had no idea that DNA might have motifs. We could slice something and do everything else to that. Wow, this is moving too fast for me. I hope it would stop. I'm just looking for anything that could stop letting it change the slides by itself, but I don't think I have that. Yes, I don't think I have that. But anyway, let's continue. So my take from this school was, I was fascinated how smart these people are and how really welcome community is, but I didn't really know what to do next. So my take was, well, if I really want to dive into that and to break into this field, I do have some prior knowledge and suitable skills that might help me to do that, but they will from technical side, so some programming, some statistics, and anything else. So I do really need to catch up on the life sciences, medicine, biology, chemistry, anything that leads you to do good experiments, which I have never done. And I also have never worked in a vet lab, right? So I have never done anything with my hands rather than writing and typing. So I decided that the best way for me would be just to join the master program that actually leads you into bioinformatics and help people from technical background to pick up on life sciences and people with life sciences to pick up on some technical skills, algorithms, and everything else. So this is how I end up being here and doing my researches. But talking about the life sciences and bioinformatics itself, we all know that bioinformatics consists from two words, right? So it's biology and informatics. And nevertheless, it's not only biology, it's not only informatics, but it's a basic structure that we start with. So it's a baseline. So actually, what biology means in bioinformatics? Bioinformatics is a truly interdisciplinary And as we discussed prior, it studies questions in biology and medicine while developing and applying methods in computer science, mathematics, statistics, and even physics sometimes, and chemistry as well. So you can really pick and choose what suits you better, what you like more. So it overlaps with medical, clinical informatics, system biology, and any other type of science you can name. And now let's talk about biology and all of that. I will try to do my best to make it simple, but it's not simple at all. So the bio in bioinformatics signalize us that biological question in studies, many of each could be grouped under the conceptual frame that you could see. So it's from genotype to phenotype, from left to right. And I have already mentioned protein a few times, but let's look at the frame itself. So for someone who might be not familiar with phenotype, in short, it is a set of observations and some characteristics of traits of organism. So what kind of? So I'm, let's say, brown-haired, brown-eyed, kind of light-skinned female. So this all is part of my phenotype. So it says something about me. These are my traits. And somebody of you might have blue eyes, green eyes, have curly hair, have darker skin, or something else. And actually, everything appearance-wise is coded is only in 1% of the genome that we hold. So it's very, very minor difference between all of us as humans. But nevertheless, this is some noticeable traits to explain for you what is phenotype is. And if you're not like me, you had your biology lesson not 10 years ago, so you might remember that phenotype is also equivalent of telling you what traits your future offspring will inherit by you and your partner. But this is it. This is for short introduction for the phenotype. Let's move on. So what else is biology and bioinformatics? So some examples might help us to understand better. So from left to right, let's see what do we have. For instance, sequence alignment are the sequence of two genomes of proteins that are similar. Could they be homologous? How can we find the closest homology of the gene we're studying from the rest of the database? Can I use a known function of a known gene to guide the study of my gene of interest? Can I do that? Is it allowed? Given DNA and genome sequences, how can we find genes in a vast of the genome? How can we compare similarities in a two-whole genome and reconstruct the evolutionary history and the process itself? How can we identify the genomic regions in methodology did? Well, as difference at the level of RNA expressions, we often define which gene are differentiating expressed between two organisms and tissues, between tumor and normal tissue, or between two different developmental stages. So when we talk about expression, this is something meaning that we could find any answer from the gene or certain part of the genome to correspond to something that we're looking for. So for the fourth part of here, you can see the numbers. At the level of proteins, how can we identify the expression from mass spectrometry data? Mass spectrometry is one of the tools or methods that allow us to catch the difference upon the mass of something that we are researching. So proteins do not exist in linear form in nature, by the way. And instead of that, they're folded in a beautiful 3D structures like the one behind me. And this was actually my master thesis project. So nevertheless, we now know that we could identify the differences by sequencing, the mass spectrometry methodology, and we also know that the basic and the most essential part of our organism as proteins, they're not linear. They are actually 3D structures. And you know what the problem with any 3D structure? It's very hard to analyze. It's very hard to find, to predict, and to analyze in any other setting. It's just, if you would like to give me an analogy, so working with stable data for data scientists is very simple. So the simple regression is simple because it's simple regression. It's good with trends, but it's not complicated. But working with pictures or videos that you have to cut in frames to give a picture, it's way more harder. So this is how you get it. So how could we predict a three-dimensional structure of protein from the sequence? Actually, it's very, very hard. And we don't know that yet exactly. There are some calculational methods that help us to do that, but it's never 100% true. And also it requires us to have a very powerful computers, and not only computers, but databases and any other structure that you would need. And infrastructure for that would be just very, very expensive. So molecules do not exist in isolation as well. And instead, they form a complex molecular network. So you see, they're not only networks in computer science, they're also networks in nature. They actually were the first one. How can we construct a protein-protein network, transcriptional regulation network, metabolic and network, or any other network that comes with nature? What are dynamic features of these networks? How do we look for them? How do we identify them? And how do we even understand what do they mean? This is when the virtual cell simulation comes in. Furthermore, with this data and researchers, how can we simulate a virtual cell simulation? This is a little bit the next stage of that. But last but not the least, how do you compare different people's genome and use population genetic approach to compare the genome of somebody ill and somebody healthy, and identify the gene mutation that cause the disease? How do we exactly do that? From these examples, we can see that there are many interesting and important questions of science that wait bioinformatician investigation, and this question are waiting the broad range of many specialists to come in. And the most important part is there is not only biologists that we need to invite, we also need to invite a mathematician, statistician, somebody who good with simulation models, someone is good with computers. So there are many specialists that we need to invite to this research. And also all of these scientists and researchers and workers or whatever, they need to understand each other. So all of them need to know at least something in another field, right? Or if they don't, how they would even discuss their investigations, right? So this is another topic. Okay, and wait a second, I need to reopen. Okay. I think we should continue. Yep, let's go from there. So, what else? Now we have an informatic part of bioinformatics. This is also very exciting. And now let's look from another angle. Completely different. From left to right, again, we have a data management, data compilation, data mining, and modeling and simulation. Some of this I have already mentioned, but let's dive into it. So informatics signalized that informational part and computational methods that we need to manage, search, and to analyze the data. So the theme runs from data to discovery. So the storage of gigabytes or even megabytes of metadata should be stored and then prosecuted. Just imagine your laptop, just like mine, probably have several gigabytes of hard disk, which is very typical one. However, just one set of our experiments rates 1 or more terabyte of data. So this is a little bit intimidating. What should we do? This is too large to fit into your laptop. So managing such big data requires advanced database systems, and also analyzing of noisy big data requires the development of algorithms and software tools that would guide you through that process. And what kind of process? So we tend to make everything open source as bioinformaticians, which actually helped a lot of scientists across the world to reach out to each other and to work together. But nevertheless, don't forget that the algorithms that we use as bioinformaticians are actually mathematically based, right? So they could also be used in any other disciplinary that you name: chemistry, trading, marketing, everything else. So it's not just for bioinformatics, but for many, many other fields, and mainly, of course, for life sciences, but I would not stop you there. And finally, when we do know how to do data mining and storage of our data, we finally, by integration of data and tools, we can build predictions. We could build the model of biological systems. As more and more high-proof technology are used, and more in life sciences and life science development with the technology, as you know, we essentially need it, there will be more and more numerous opportunities for technologies in bioinformatics. Because we do not start there. So what do we need for this challenge to happen and to be accomplished? To meet this challenge, a new breed of professionals, as I said prior, need to be trained and hired. So we could call it a collaboration or just a similar and a pure working process. But initially, we need many people with various backgrounds coming together to solve many biological-based problems, to simulate the system, and then to make any sense of the results. And it's not only of biology, medicine, and technology. There are also many legal questions that would arise. There are also many ethical questions that would probably arise when we talk about personal data and many other things. So it's not that easy as that might seem. As I mentioned, bioinformatics and any other life science that you call name of the science and informatics or something analysis, it's not just these two, and obviously there would be many more fields to be involved. But all right, I hope you do have at least a slightly opinion about what bioinformatics does, what these people do, what also are researchers for life sciences. But who are these people, right? So we talk a lot about computers, software tools, algorithms, but what else? So actually, there is no question about why these people are in so high demand. So information is the oil of the 21st century, and analytics is a conversational engine. This is very true. And what makes it true? Because we do collect more and more data, and we need people to understand this data, because the raw data does not make any sense, and it does not make any profit to any of us. So if we try to summarize what we have already discussed, we have talked about the question the specialists try to answer. Even through each year, we have new uprising and undiscovered topics, right? We already talked about the bio part and information informatics part in bioinformatics. We also slightly covered life sciences as well, but don't let proteins fool you. That's my favorite saying. So any of these discussions and questions that we discovered, they're not only biological based, right? And it's not always about the production, but it's also about the medicine and providing the best treatment for your patient and also just for well-being as a human beings. So it's biology, chemistry, medicine, and pharma as well. We also talked about some up-to-date tasks as cancer research, diabetes research, as well as we covered some technologies that are involved with databases and algorithms. These are the main ones. But what still bothers me, as I hope bothers you as well, we still have no idea who are the specialists are. How do they become them? And thus, from now on, I would try to focus on people more rather than what they do, but let's try to make a portfolio of a life science analyst, as we can say it so. So let's run from the issue number one, that struggle that we would face. There are massive job titles. So usually when you call someone an accountant or a basketball professional player or a teacher or a musician or anybody else, you really know what this title means, right? And of course, there is also some confusion in modern job titles. They usually align with software development area, but anything in the edge of science and the software would be just as confusing as, if not much, if not more. So from my experience that I have witnessed myself, there are just a few types, actually 5, that jobs or job titles that you would have or you would look for in future to get your job. So what are those? You could be called an analyst, researcher, specialist, statistician, or something unique. With something unique, I would come with something with bioinformatician or biostatistician, something that already established. Well, bioinformaticians, there are, of course, all of them. There are researchers, analysts, specialists, statisticians, biologists, or chemists. So there are many, many things, but just to name a few of them. And so here in the right, you can see, so there are a few areas that you might work in the future if you choose to be something in between the technology and life science. You can work as bioinformatics, in cheminformatics, biostatistics, medical statistics, healthcare, data analytics, biotech, clinical research, health information management, computational biology, chemistry, genetics, genomics, whatever it is, healthcare analysis, drug discovery, electronic healthcare records. So there are many, many various of fields or subfields that you might want to go to. And also it will be not only dictated by your choice, but also by your primarily set of the skills that we also would cover a little bit later. So let's go further. All right, so what does a healthcare data analyst do? So if we have been focusing mainly on the research topic like cancer or diabetes, and we look for genomes and we sequence and we do research and we do develop some algorithms, but if there's something more industry focused, of course there is, and it exactly healthcare data analyst. Why do I speak about that? Because not everyone is a science person, even though they work in the edge of the science and technology. Sometimes you do want to see the result of your research while you are alive, right? This is actually what I do. I'm more practical, so I want to know if I helped somebody or didn't manage to do that during my working week and not in 10 years. And I want to be able to pursue my research work in my lifetime, right? So if you're also looking for something more practical, that would be a healthcare data analyst. And do you mind, this is a very broad naming as well. So every naming, every title, job title would be very messy, but we have to call it somehow, right? So what if we talk about something more industry focused, right? So let's see what healthcare analyst does. It's not a simple answer, and work may differ often depending on who is employer. So there are several types of employers. So first type is hospitals, pharma companies. Sometimes it is stayed in genome statistics, collaboration, or in modeling different hospitals or providers. So because there is no just one healthcare data analyst role, the day-to-day work for this job can take a little different from one role to another. But there are just four main categories that I would like to talk to you today. So the first one and all of these categories will be basically divided by your employer. It's a healthcare providers. Who are they? What are they? So basically they include primary care physicians, hospitals, long-term care facilities, or any living facilities, or urgent care patient-facing roles, basically. So these are all roles of very patient-facing. What does it mean? You get a lot of clinical data. So this type of analysts often work with clinical data to help improve patients' outcomes. And they also analyze patient data much in the same way as analysts in any other field will do. So if you're a marketing analyst, financial analyst, you do practically the same thing, but the meaning of your data is different. That's the key point. So the data itself is specific to medicine, while many systems and informatic tools are very similar to any other field. So if you know what CRM is, it's much like any medical clinical system. So it just counts of data. It also counts of someone who uses data. So it's very similar. Who else might be your future employer? It might be suppliers, supplier companies. Who are they? So suppliers cover a wide range of ground and groups, but some suppliers are software by nature, as medical software systems that we talked about previously. But suppliers also include the pharmaceutical industry for developing pharmaceutical drugs in production or to cure the disease. And it also involves some device makers, which are essential for medicine. And it could range anything from implanted devices to diabetic testing cups or bandages. And anything that may be used in a treatment or related issue or prevention is also would be considered as a device. And don't forget about some kind of MRIs, scans, etc., etc. Every technology-based device that you may see in a hospital is actually a device supplier. So the supplier category is probably the broadest one, and there are so many niches. And in general, their analysis tend to focus on clinical data as well, but there is also tracking supply, collecting data for improvement, recommendation systems, and many, many more. So if you have ever worked for any other systems, so you would know. Clinical data analysts fall into this category as well, and they mostly work on evaluation of the performance of something. It might be a drug or treatment or anything else. The third category is insurance industry. So it's not that broad and common in Russia, but I would say that is much more prevalent in the United States than other countries. It's just because of the systems of the healthcare, while in the United States, they are all covered by insurance companies. Here, it's more universal. So healthcare analysts in this role can look at variety of data, right? Everything from general trends about patients who are insured, increasing or decreasing of health concerns, analyze specific patients as well. So it's one-to-one work, very similar to the naming price range changing, right? So they try to make a right price, not only the price, but the price is very important for the company. So the company obviously looking for minimizing their expenses or costs for treatment. And sometimes it does align with patient interests, but not always. And I'm sure you can name many examples of both situations. And and there is a last but not least category, that would be policy and regulatory. So for instance, in the USA, it would be FDA. For Russia, it would be the General Министерство здравоохранения. So these roles typically concern themselves with understanding of picture of public health in whatever area they're covering. So it might be public health, public food, or anything else. So they work often factors into public health programs and hospital, or providing rating of the hospital. So their outcome is basically the system that we're using. So we can learn what health concerns are increasing or decreasing. Is it smoking, obesity, diabetes, cancer, etc., etc.? So the essential question that comes after that we have talked about this is, do you need a background in healthcare? A lot of this would depend on specific area you work with, like having a knowledge of that specific for healthcare or care supply or whatever is always an advantage. But in some roles, it's going to be less critical than other, which is obviously, as I mentioned, some of the roles are very similar to working as data analyst in any other field, and then you don't really need to understand the data itself or know any specific detail of the condition of this disease, while working with clinical data would probably require some prior experience in medical providing services. If you worked as doctor, nurse, or you have a prior education like I did or many other people have. So there is no unique answer for that. And hopefully you can look through this type of screenshots of what actually a bioinformatician need to do and what they are required to do on their work. And it also varies a lot. Some jobs do require you already to know and have a practical experience, hands-on, some tools. So you just had to work with them. And also tools varies a lot from what kind of data you work with. Is it Hi-C? Is it single cell? Is it anything else? Some of the jobs also require you to know some basic systems like Power BI systems to work with the reports. You also need sometimes to work in a wet lab, so you need to know some practical wet skills, even though it happens to be rarely, but it still comes across if you look for a bioinformatician or any healthcare data analyst role. As well, they might require you to know a lot of mathematics and computer science. Some of them would require a basic knowledge of the Excel. So it really depends on the work. So if you want to break in somehow in this area, I have a few slides for you. So what about formal education? If you don't have a prior experience in any of healthcare industry or medicine or biology, and maybe you just were not interested in that before. So if you're a student and you have an opportunity to go study, there are just a few universities and programs that I found and which I have looked for myself some years ago. You can join them. There are also many courses that are available online. There are also summer schools, so you can get your hands-on experiences to see if that area even matches your expectation. And I would call myself a typical path to bioinformatics before that. And I would say that typical ways you get a bachelor's in mathematics or this life science, and then you gain something that you lack in knowledge during your master's, and many of the people who decided to work on the edge, they do pursue to have a PhD in these areas, and there are many PhDs in cancer research, bioinformaticians, and many related life science compiling with computer sciences. So they usually do work between two departments. So for instance, in the High School of Economics where I did get my master, the program is called Data Analysis in Biology and Medicine, and we did study a lot of chemistry, biology, medicine, experimental type of subjects, but it was based on computer science faculty. So you see how it goes usually. And what about skill set and knowledge, what you need? So as I said, it varies from position to position, but as far as I can judge, and I have been working a lot and also on my thesis, and I worked in labs, and I worked as a data analyst in other fields, and now working with artificial intelligence in production. I would say that knowledge of the technical skills are more important than the area of life science at the beginning. Why so? Because there's so many parts of life science like genomics, proteomics, metabolics, transcriptomics, epigenetics, public health, computational science, microbiology, micro everything, clinical everything. So there are too many to choose from, and you would never know where you would end up unless you specifically know that you would do this specific field. So you really need to know how to code in Python and R. Why R? Because this is just a very typical language for bioinformatics. It just happens to be historical, and the R contains some of the libraries that are not yet translated to Python. So it's better and more beneficial if you can know how to use both. Also SQL for accessing the databases, and I told you multiple times that databases are essential for any work in life science. And command line, just because many tools that are used by bioinformaticians, they started from command line. So for mathematics part of that, while we discuss the programming languages, actually you have to evaluate your results. And how do you do that if you don't meet your patient by yourself? So you have to know the statistics, machine learning, and any other tool of artificial intelligence. So I would say that statistics is one of the most essential parts. You need to know at least what is p-value, what is e-value, what is first hypothesis, how to test the hypothesis, and what are the probable outcomes. My favorite question, the one that I enjoyed a lot, is that if you test the drug for your nose, and as you know, there are two holes in your nose, and it worked in one hole but didn't work in another. Is the drug okay? Should we let people use it? So how we did end up with such results? Just because people who evaluated results, they didn't know about Benferroni. So when you test something multiple times, you have to do some additional testing and evaluation. So the outcome of that, your results should be universal for the whole body, preferably, or for the vast majority of your patients that you tested. So you need to know what's the test, train, and other sets of data. So there are many things you have to know, and I know it's confusing, but I hope you can manage with that. An area of knowledge is actually the life science that you choose to pursue. It might be very different, and even though in one particular science there are many sub-scientists that would work on only this bacteria or only this type of data. Some of them do meet patients and people, some of them do not meet them. So it just depends on what feels comfortable for you. And as well as there are many already written tools that make our lives extremely easy I'm joking, just a little bit easier. So there are many tools for finding short read alignment, global alignment, variant calling, working with FASTQ data quality, and many, many other ways. So it's better for you, and it would come naturally as long as you work in the field, that you have to know how to use and what's inside, what's the sense of the tool, right? So you cannot only blindly respect and blindly believe in the results. You always have to check it upon the common sense at least. All right. And this is all for today. I hope I didn't make you sleep, and I hope you did enjoy at least some of my conversations. So if you have any questions to be asked, it's time to do that in the chat, or you also have, I think there is a question that I think there's a button that you can push to make a question available. So do whatever suits you. The presentation will be available later, and as well as the recording of all of this. And you also have my contacts to, if you would like to approach me personally later. And my personal recommendation, there is International Laboratory of Bioinformatics in High School of Economics. So if you're interested and you're just about to start, so look for the events, look for the open job positions. So enjoy yourself, and I hope you will have a good life and a good research in bioinformatics. And I hope you will have a clear data in your life sciences. So that's it from me now. And now it's time for you. If you have any questions, I'm here to answer them. Thank you for your attention. Thank you for coming so late to listen to me after the work. I know it was a hard day, so I do appreciate this a lot. And one more time, I'm very sorry that it was so chaotic with the presentation. Unfortunately, I didn't have any prior experience on using that, so I'll just hope that next time I will do better. All right, let's wait, let's say 3 minutes. If somebody will come up with a question, or somebody would be interested in reviewing or asking for any other experience, so I will be here. There's a question about if you go for PhD in Russia, what kind of PhD you will obtain. It really depends. If you go for neuroscience, you might get a medical PhD, кандидат медицинских наук. If you go for anything with cancer research and anything else which would lead you to more biological question, you would be biology, so кандидат биологических наук. Rarely, but it's also possible if you're based on technical faculty and this is just a minor lab, or your thesis advisor is looking for somebody in bioinformatician, you could become a PhD in I would say like mathematics or technical science. So it would be probably кандидат физико-математических наук, but this is a very rare occasion. I would more likely looking for biology or medicine. Yeah, I also thank you very much once more time for coming here, and I hope you had a great evening after that. And I hope you like life sciences just like I do. There's one more question about are there any serious opportunity in industry in Russia, or most of them are abroad? So it depends on what do you consider a very good opportunity, but of course there are many of them in Russia. So there are many labs that do research primarily on the edge of technology and biology. So many of my group mates from my masters do exactly that they work as bioinformaticians or life science data analysts. And also I do believe that in future many smart hospital systems would come to the hospitals, so they would need data scientists with a knowledge and a background of medicine and biology, and I could also call it data analyst in life science. That's what actually is my ideal job would look like. And as well as you could also go for PhD in Russia, aspirantura. So there are many things you can do if you look for opportunities. But I would say, without a doubt, that abroad, this specialty would be a little bit better in salary, but it's always a, a question of how do you sell yourself to the employer. But yeah, so I hope if you do look for opportunity, you definitely will find one, but probably here you would need a little bit more experience, but it also depends on the lab and on the people that they're hiring. Yeah, thank you, Nikolai, for the question. I also hope you will have a wonderful evening. Yeah, let's be there. I will be there for, let's say, 3 4 minutes for you. If you still hesitate to type an answer, type a question, it's your time to do that. I'm here to answer all of them as much as I can. But don't forget, if it's not your thing to do to ask me publicly, you can always approach me by the credential that I put in my presentation. Feel free to approach me, and you will also be able to download the presentation later. All right, let's say 1 more minute. So if you have anything to say, to ask, or anything, it's your time to shine. Okay, seems like everyone has asked everything they were curious about, and I'm over with my presentation. So it seems like the right time to say goodbye. One more time, thank you for being here, and I hope you have a good evening and a good month and good everything. So, bye bye.